Good afternoon. So I'm Shricharan Senthilkumar. Today. I'll be just speaking about the post-harvest losses of horticultural products. So before entering into the presentation, so let me introduce myself. I did my undergraduate in agricultural science from Annamalai University. Then my master's in the field of plant physiology at Indian Agriculture Research Institute, New Delhi. During my master's, I worked on abiotic stresses, especially the high temperature stress in wheat by the mechanism of maintenance of stake green rate. And currently, I'm about to join as a senior executive in research in a startup called VGrow. There, I'll be working on the post-harvest losses of especially fruits by developing like innovative solutions and formulations, optimizing the supply chain mechanisms through improving the reducing the post-harvest losses or by increasing the shelf life of fruits. So to begin with the presentation today, horticultural products has fruits, vegetables, flowers, and foliages. So fruits are packed with rich in vitamins, sugars, and they're also a key component of a balanced diet. Coming to vegetables, they are rich in essential nutrients and also contribute to the overall health. So we take it daily in our diet. Next part is the flowers and the foliages. So both the flowers and foliages are used for the purposes in the floral industry, landscaping, as well as for the interior decorations. So, coming to today's topic on post-harvest losses, this is a statement given by our Honorable Prime Minister. Food security is a challenge. So, we must work on cutting down the post-harvest losses. So, as we all know, horticultural products are perishable in nature, especially the fruits and vegetables are perishable in nature, maybe because of their high moisture content. So, the main common approach to reduce the post-harvest loss or by extending the shelf life of the commodity can be storing it under the refrigerated conditions. So, when we speak about the cold storage in the India, its ownership lies maximum of its ownership lies with the 95% under the private sector, with only 2% in the public sector and the 3% in the cooperatives. So when we see the status-wise cold storage facilities in India, mainly in the southern part of India, Chhattisgarh has only 1% of the cold storage, followed by Tamil Nadu, Telangana, and Odisha. So FAO has also said that in India, around 40% of the fruits and vegetables, which is around $8.3 billion, are lost every year because of the post-harvest losses. So loss in fruits and vegetables is quantified about 30 to 35%, which causes an economic loss of about 2.4 lakh crores annually in the year 2017 to 18. And the next statement is also suggested by FAO that 40% of the fruits vegetables is like about $8.3 billion in India is lost annually. So again, because of the lack of proper storage and transportation facilities in India, about 20 to 40% of the tomatoes are getting wasted. So the three main reasons for the post-harvest losses are the harvesting techniques, the poor storage facilities, and the poor transportation facilities. And speaking about the flowers and foliages, approximately 30 to 40% of the gets wasted because of the lack of proper post-harvest management. So why is the need to address the problem? The first point is because of the food and nutrition security. So according to an Food and Agricultural Organization, approximately 1/3 of the world's food production, which is produced for the human consumption, is getting wasted. Next is on the economic impact and the farmers and in the industry. World Bank has estimated that economic value of global post-harvest losses in fruit and vegetable is around $150 billion annually. Next, speaking on the environmental sustainability, the impact of post-harvest losses include the wasted resources like water, energy, labor, land, manpower, etc. Additionally, when decomposing this organic matter in the landfill also contributes larger percentage to the greenhouse gas emission. Finally, the global trade and the market taxes. So International Trade Center reported that reducing the post-harvest losses of the product can significantly enhance the export competitiveness in the developing countries with respect to the horticultural trade. So entering into the physiology of the product, how it is getting ripened and it's damaged, or like how it is getting senesced and lost during the post-harvest stages. Any horticultural commodity will undergo the phases of from unripened to the ripened and followed by the senescence phase. So when a crop, I mean, like when a product is under unripened stage, it is characterized with high cellulose content and low sugar content. So the advantage of this stage is that shipping can be done with no minimum damage, and also will have longer shelf life. But the problem is that it has the lower quality as well as the low cost. So when we speak about the ripening stage, it is characterized with moderate cellulose content, low starch content, higher sugars, and a high titratable acidity. And the problem here is that shipping problem because of the mechanical damage and the shorter shelf life. But speaking of the positive aspect, it has high flavor and nutritional quality and also higher cost. So coming to the third aspect of damage, when there is a little cellulose content, it refers to the cell wall breakdown. So the sugar contents get altered and the flavors gets improved. So this reduction in the cell wall content or the permeability of the cell wall is susceptible or like attracting the pathogens like fungal as well as the bacterial infections and causes damage and makes it low quality and also inedible. So this cannot be consumed, and also post harvest injuries on the end of shelf life is higher. So this slide speaks about the climatic and the non-climatic fruits. So climatic fruits are those fruits which can ripen even after harvesting it from the plant. So normally this fruit is characterized by increase in respiration rate as well as the ethylene production. So ethylene is a basically a gaseous plant hormone which is involved in ripening of fruits. So while the non-climatic fruits are those like fruits which cannot ripen when they're just removed or detached from the plants. So if a non-climatic fruit has to be ripened, they must be intact with the plant. Then only it can be matured fully. So on the aspect of growth curve, the growth and development of any plant or any organism will follow a S-shaped sigmoid exponential curve. Normally, under the growth and development phase, it increases exponentially and reaches till the physiological maturity. Here, speaking about the aspect of fruits or the vegetables, at physiological maturity, the growth is maximum, and thereby there has a steady phase where it reaches the ripening stage, then the aging or the senescence phase. While in the climatic fruits, the rate of respiration is found to gradually reduce from the growth and development phase till the maturity, that is the physiological maturity. And farmers harvest the crops or the product at the physiological maturity stage. So during that time, after the product is harvested from the plant, they have an increased rate of respiration, also followed by an increased rate of ethylene biosynthesis. So these two mechanisms activate the plant processes which is involved in the ripening. When we speak about the non-climatic fruits, there is a gradual reduction in the respiration rate till the end of the aging or the senescence. So it is confirming that only when a product or the fruit is intact with the plant, a non-climatic fruit can reach the maturity and lead to the aging stage. So this non-climatic fruit will also produce only a little or negligible amount of ethylene, and it also responds poorly when this ethylene is exogenously applied to it. So this is the major difference between the climatic and the non-climatic fruits, and with respect to their physiological mechanisms governing their ripening stages. Now let me enter into the topic of maintaining or improving the shelf life by reducing the post-harvest losses. So this slide speaks about the edible coating. Edible coating is nothing but you coat a material which is edible for human consumption, with the materials like polysaccharides like starch, cellulose, cytosine, pectin, alginate, along with essential oils with antimicrobial properties, also with other plant extracts which are rich in antioxidant properties. So this coating emulsion can be applied to the fruit or the vegetables by different methods like spraying, dipping it, or painting it, or spreading the emulsion above the surface, or by multiple layer of coating. So the major impact of this is to prevent the internal physiological process from the exposing it towards the outer atmosphere. So thereby it act as a covering. Thereby the rate of gases exchange like carbon dioxide and oxygen level within the fruit and the outer atmosphere can be cut off. Hence, the respiration rate can be reduced. Also, the endogenous production of ethylene rate can also be reduced, thereby maintaining it for longer duration. So, this image speaks about improving the shelf life of strawberries. So, in the control, it is seen that only strawberries harvested remain in edible state only till the end of 10th day. So while they have applied three treatments, well, T1 is coating with pectin of 3%, T2 is pectin of 3% with lemon essential oil of 2%, and the treatment 3 is the pectin of 3%, lemon essential oil of 2% with lutein of 10 millimolar. So this suggested that the T3 treatment shows to improve the shelf life by reducing the microbial attack till the end of 31st day. So edible coating is one of a upcoming promising technology, like a practical strategy to improve the shelf life of horticultural products. Next, speaking about the modified atmosphere. Modified atmosphere is nothing but like you wrap the product with materials like polymers, like polypropylene, or the biodegradable polymers like polylactic acids. So the mechanism governing here is that the modified atmosphere oxygen concentration is only about 3 to 5 percentage, which is maintained around the fruit. So this 3 to 5 percentage of oxygen concentration is found to reduce the level of respiration rate, and also the enzyme activities, also the biosynthesis of ethylene hormone. So this results in delayed in ripening. Uh, Also, the changes regarding the enzymatic browning also maintains the weight loss and the retention of the firmness, total soluble sugars, as well as the titratable acidity. And the carbon dioxide concentration is maintained around 5 to 7 percentage. So this higher concentration of carbon dioxide around the fruit makes the fruit less sensitive to the ethylene synthesis as well as the ethylene responses. So a higher concentration of carbon dioxide can also help in controlling the fungal or the bacterial infections and alter the intercellular pH of the cell membranes of the microbes. So this modified atmosphere around the material is also a promising strategy which can be adopted during the supply chain processes of the fruits and vegetables. So this tells about the mechanism. This is a case study in case of Oriental melon. When the melons are packed with modified atmosphere, they have shown to improve the shelf life when compared to the control. This also reduces the firmness, increases firmness, and also reduces the chilling injury by reducing the rate of lipid peroxidation and also weight loss. So this is an image of controlled atmosphere where chamber is fitted with the carbon dioxide scrubbers, also with the ethylene scrubbers and nitrogen cylinders. So this chamber is temperature controlled with along with the humidity. So it is a controlled environment where we improve the concentration of the carbon dioxide by reducing the level of oxygen by supplying the nitrogen gas. So this can also improve the shelf life or the storage life of the fresh produce of the horticultural crops. But the major disadvantage of this is of higher cost, as well as it cannot be used for long supply chain or the transport facilities. Now speaking about my area of research interest is on the plant growth regulators or the plant signaling molecules. So the first is about the carnation flower. Actually, this carnation flower is treated with different concentrations of melatonin. So this melatonin concentration of 0.1, 0.01, 0.1, and 1 micromolar has found to improve the shelf life of the or the vast life of this cut flower carnation. So when comparing all this treatment, 0.1 micromolar concentration of melatonin has found to increase the vast life of this carnation up to 10 days when compared to the control. Now speaking about this cut flower zebra, this flower is prone to chilling injury as well as also characterized by low shelf life. So the figure A represents that under 8 degrees Celsius, the stage 3 of the flower is seen of a curved neck symptom near the peduncle of the flower. So this 1.5 degrees Celsius storage is found to increase the chilling injury damages to the flower. While the B figure shows that 2 times 3 time treatment of GABA. GABA is nothing but gamma aminobutyric acid. Along with the next treatment is the spermidine. So these are polyamines and also naturally present in the plants. They are like natural signaling molecules present in the plants. They have found that abiotic stress mitigation effect has been found in these two signaling molecules. So under 8 degrees Celsius storage, application of this GABA has found to increase the shelf life when even when compared to the control as well as the spermidine application. And while storing it under the 1.5 degrees Celsius, that is chilling temperatures, it has also found to reduce the effect of chilling injury in the GABA spray. So GABA is also a potential molecule along with melatonin in the previous case to improve the vast life of the cut flower when compared to the control under normal stored conditions. Coming to the role of ozone in improving the shelf life, ozone is found that it has an antimicrobial agent and anti-disinfectant properties. So the above images shows that tomatoes are found to increase the ozone treatment in tomatoes has found to improve the shelf life up to 30 days, while in 10 days of cucumber, 7 days in case of bell pepper, and also 7 days in case of strawberries and 18 days in case of beans. So exposing it of 130 to 150 ppm on an average of for an about 3 minutes has found to improve shelf life in all the above commodities. Now speaking of ozonated water, this ozonated water cleaning the product with the ozonated water has found to improve the shelf life of strawberries about 8 days when compared to the control, that is the tap water. So these are some of the studies conducted in our lab. Like we have worked on the cut foliage dracaena. This is used as an ornamental plant, also serving as for an aesthetic purpose in various functions and ceremonies. So we have three plant growth regulators along with one of our formulations called Biomax Plus. So this Biomax Plus is a crafted formulation especially for this dracaena cut foliage with different bioregulatory molecules, plant signaling compounds, plant nutritions, and it is shown that this Biomax Plus formulation has overthrown the individual effect of plant growth regulators and also the water as a control. So now we are in the process of developing the Biomax Plus solutions for different commodities of fruits, vegetables, foliages, as well as the cut flowers, and also in the process of patenting it. Now we have also worked on improving the shelf life of chilies, especially the green chilies. Our product like Biomax Plus, especially crafted for the chili, has found to improve the shelf life of chilies from 0 to 30 days when compared to the control. So we have applied this Biomax Plus product by foliar spray on the product, like surface coating it about 3 minutes in the dipping. Next, let me enter into the molecular mechanisms governing this improvement of shelf life or reducing the post-harvest losses. So abiotic factors, or we call it as environmental factors like temperature, light, water, nutrients, and other stresses, can directly influence the epigenetic modifications like DNA methylation, chromatin remodeling, and also influence the non-coding RNAs like small RNAs, microRNAs, and long non-coding RNAs. So these epigenetic modifications, along with non-coding RNAs, can positively or negatively influence the transcription factors like MEB, NAC, MADBOX, and ERFs. And also the plant hormones and signalings like ethylene, ABA, sucrose, and nitrogen dioxide and calcium signaling molecules. So this transcription factors, along with the plant hormones, this epigenetic modification and the non-coding RNAs, can directly regulate the ripening genes like lycopene B synthesis, AMY, AAT, LOX, PL, PG, and PME, and so on. So these genes regulate the biochemical pathway which is involved in the pigment biosynthesis, vitamin biosynthesis, cell wall or the starch degradation, sugar transport, and others in both the climatic as well as in the non-climatic fruits. So now we are in the era of genome editing. So genome editing with respect to our area of research can be used to improve the shelf life and also the texture of the fruits or vegetables. So this is a research where they have found that the loss of function of this long non-coding RNA 1459 is found to delay the tomato fruit ripening. So when we see the figure, this is the wild type tomato. DAP represents date post anthesis. So it is found that the tomato is completely matured by the end of 50 days post anthesis, while the long non-coding RNA 1459 mutant varieties, or the loss of function mutant of this long coding RNA has found to improve the shelf life or delay the tomato fruit ripening in both the cases. So this is all done with the use of CRISPR-Cas9 genome editing. Next study has found that Solanum lycopersicum NAC4 is involved in ripening of tomato fruit. So this is the wild type crop, wild type fruit, where at 44 days post anthesis it reaches the complete maturity, that is the red ripening stage, while overexpression of this NAC4 transcription factors has found that even under 36 days post anthesis, the tomato fruit has started in the stage of breaking stage, and it is increasing the rate of tomato ripening in both the cases of overexpression of this NAC4 transcription factor. While NAC4 transcription factor knockout has shown that even at 44 days post anthesis, the tomato is in orange color and not completely fully ripened. So this NAC4 involves in the ripening of tomato fruit, and this mutant has shown that uh, it has reduced. Like it has reduced the rate of tomato ripening and also has increased the rate of shelf life. So here it is shown that the NOR like one is no ripening like one, is a new positive regulator of tomato fruit ripening. So TRV is tobacco restil virus. So with this TRV control, it is shown that BR is the breaking stage, B plus 5 is the breaking plus 5 days, and B plus 5 the cut section of the tomato. It is seen that after breaking stage of 5 days after the breaking stage, the tomato is completely orange or little reddish in color in the control. While NOR like one mutant or the loss of function of NOR like one mutant has found that even after the 5 days after the baking stage, tomato is half ripened, and this shows that is the positive regulator of tomato fruit ripening. So the next study sees shows that in NR in non ripening like one has found that at even at 55 days post anthesis, the tomato is in the stage of breaking stage, while compared to wild type, this breaking stage is in the stage of 38 days post anthesis. So this NOR like one is found to be a positive regulator of tomato fruit ripening, which has a different aspect to work on for improving the shelf life and also reducing the post-harvest losses. Next is the Solanum lycopsychum grass 4 transcription factor. This transcription factor is found to accelerate the fruit ripening by regulating the biosynthesis of ethylene genes. So the wild type is found that till 48 days post anthesis, the tomato fruit is in orange color, while the overexpression of this GRAS4 transcription factor has found that at 42 days post anthesis, the tomato has found to be in the fully ripened stage. So, mutation or the genome editing using on the specific target GRAS4 can reduce the fruit ripening by regulating the ethylene biosynthesis gene so that it can improve the shelf life of the tomatoes. So these are all the few examples where CRISPR-Cas9 genome editing has been playing a major role in improving the shelf life as well as the texture flavor of the tomato with respect to reducing the post-harvest losses. So to conclude my presentation today, the critical global issues has to be addressed. This post-harvest losses in horticultural products poses a significant global challenge, and this also impacts the food security, economic stability, and also the sustainability in agriculture. So multifaceted solutions to address the post-harvest losses are required. So this involves the encompassing the innovative technologies, sustainable practices, the regulatory support from different sources, and the collaborative efforts. Laboratory research contribution, of course, plays an important role in developing solutions to mitigate post-harvest losses. These provide an insight into the physiological, environmental, as well as the technological factors or the mechanisms influencing the product quality. Then the global initiatives like led by private organizations, food and agricultural organizations should focus on reducing the post-harvest losses by collaborating and implementing best practices with supporting technological advancements. And finally, it is a collective responsibility from the researchers, policymakers, industrial stakeholders, and the communities to ensure the scientific advancements integrating into the practical agricultural applications to find more sustainable and resilient food for the future. Thank you.
Thank you, Mr. Shricharan. I will now take up some questions. Just give me 1 second. Okay. So the first question is, how can small-scale farmers integrate efficient harvesting techniques into their daily practices?
To answer this question, speaking about the impact of post-harvest losses with respect to the harvesting practices, I have come across a case study where chili farmers harvest the crops, or I mean, they harvest the chilies under different days after the flowering. So when the chili is harvested along 11 or 12th day after the fruit formation, it has found that that stage of harvest has found to increase the shelf life of chilies when compared to harvesting it on the 15th or 16th day. So even the stage of harvesting and the time of harvesting also plays an important role where farmers can adopt it in their field to improve the shelf life of the crops, the products.
Okay. What on-farm practices can small farmers adopt to minimize post-harvest losses in horticultural crops?
So like actually, I have answered it already. Like harvesting technique or the stage of harvest also plays a major role in advancing or reducing the post-harvest losses. So the correct time of harvesting and the correct stage of harvesting can only be a simple solution for the farmers. And after the harvest, products, there are commercial products in the industries already available, and also we are coming up with the new formulations like Biomax Plus, which is completely natural. So just coating it with the formulation or just dipping it in that formulation can also extend the shelf life of the commodity under normal stored conditions. So farmers can adopt these technologies like simple and practical strategies to improve shelf life with respect to their levels.
Okay. And what affordable and practical storage solutions are available for small farmers to preserve the quality of their horticultural produce?
Uh, with respect to speaking with the quality and the post-harvest losses in the small scale farmers, they may not be may be accessible to the cold storage facilities. So it is like how we manage it during the harvesting, like without any damage, or the stage of harvest, cleaning it with proper water, or exposing it to little amount of sunlight, or the disinfection practices can help small farmers to improve quality as well as the shelf life of the product.
Okay. And how can farmers adopt traditional stage storage methods to reduce post-harvest losses?
Speaking about traditional post-harvest storage methods, in case of onions, like they stack up the onions with some airflow in between them. So this method helps the farmers to improve the shelf life of onions, and also in case of potatoes. So the major post-harvest problem in potato is the potato greening or the sprouting of potatoes. So there are some plant extracts with this antimicrobial properties, or the some kind of properties which can influence the plant mechanisms which involved in like sprouting of potatoes. So adopting of these technologies can help farmers to improve the shelf life as well as the quality by adopting the normal traditional storage conditions.
Okay. Mr. Shricharan, and before I take the question, next question, are you comfortable in switching on your video?
Yeah, yes, ma'am.
Yeah, okay. And next question is, what affordable and practical storage solutions are available for small farmers to preserve the quality of their horticulture produce?
Like, sorry, ma'am, I think like it is like a repetition of the same,
Oh, okay. Yeah, sorry. Yeah.
like,
What are the correct?
yeah.
What are the common challenges faced by small farmers in transporting horticulture products, and how can these challenges be addressed?
Yes, ma'am. So speaking about the supply chain industry, especially with respect to the small scale farmers, there is always a price fluctuations in the horticulture industry where horticulture, especially the horticultural farmers face to transport their products to the customer's end. So cold storage facility is one of an option, but this cannot be accessible by all of them. So mechanisms like products like our Biomax Plus can improve farmers to improve the shelf life. Just dipping it in our solutions for about 3 to 5 minutes has found that it can improve the shelf life of chilies for about 30 days. So this is like a suggestion for the small farmers, like when there is a price fluctuations, or there is a reduction in price in the rate of tomato or chilies, they can use these kind of formulations so that they can improve the shelf life up to 30 or 35 days. So and then they can transport it, or then they can bring it to the market when there is increase in price, so that farmers can also get a better income out of that product.
Okay. And yeah, are there innovative transportation solutions that can be adopted at the local level to prevent post-harvest losses?
So actually, I am not working on that aspect of transportation or the supply chain. I am about to work on that or optimize the process of supply chain industry. But in general, adopting normal practices like which I have mentioned earlier can reduce or cut off the use of cold storage during the transport. So this may also be an innovative solution for reducing the cold storage facilities, which is necessary even during the transport of the products.
Okay. And how can small-scale farmers explore value adding opportunities for their horticulture products to reduce losses and increase income?
So value addition is one of a different approach where farmers can convert their product directly into many forms. Like if mangoes are having a lower shelf life and the commodity of mango is huge in number during a particular season, they can convert the form of mango in sort of a pickle or jellies or jams where they can fetch a larger product price in the market. So value addition is also one of a significant approach to reduce the post-harvest loss and also to increase the monetary means in terms of farmers.
Are there successful case studies or examples of farmers or communities effectively minimizing post-harvest losses that we can learn from?
I have not gone into like successful stories like that. Maximum cases I've just in the research and development line. So I'm also participating. Like I'm in the process of taking the research activities. I mean, like the lab to land process. So in the future, there may be useful results from the lab studies, which can positively or converted into a commercial successful stories in the upcoming years.
Okay. And how can collaboration between farmers, government agencies, and private sector stakeholders contribute to reducing post-harvest losses?
So this is very important because like there should be extension agencies in form of government agencies or the farmer producer organizations where they can bridge the gap between the lab and the land. So bridging this gap is very essential to bring out the innovative formulations or the development which we develop in the lab to the field of farmers. So development of policies with respect to farmers as well as the startups and the companies working in the post-harvest losses or fruits and vegetable handling is very essential to link both the lab to land processes.
Okay. And in the context of climate change, how can farmers adapt their post-harvest practices to address new challenges and risks associated with changing weather patterns?
So this is an era of climate change where we have to speak about the different aspects of climate, including the rainfall, precipitation, the high temperature, chilling injury, everything. So in case of cold, I mean like cold products like strawberries or potatoes, this climate change is posing a serious threat in the production as well as the cold storage. I mean the supply chain or the storage or improving the shelf life of the crops. Like development of innovative solutions in terms of practical strategies like using this in terms of edible coating or the genome editing can improve in the development of climate resilient varieties with improved texture, quality, as well as the shelf life. So development of climate resilient varieties with improved shelf life is one of the best option in case of climate change.
Now we have the last question. Are there regional or cross-specific factors that contribute significantly to post-harvest losses, and how can we tailor solutions accordingly?
Sorry, ma'am, can you please repeat it
Yeah,
once?
and that. Are are there regional or crop-specific factors that contribute significantly to post-harvest losses, or how can we tailor solutions accordingly?
So, like every plant has different physiological mechanisms operating inside it. So, basically, to study the physiological mechanisms, the process of ripening, the senescence, the aging process, and finally the microbial decay, need specific time and the controlled conditions to study them. So, once the physiology of the product or the horticultural commodity has been studied, then different aspects of using of products like commercial product like coating it or using of nano encapsulations, adopting technologies with the modified atmosphere or the genome editing can help to solve this purpose. So basic purpose has to be studied from the physiology of the process.
Okay. So yeah, there is a question from Sivabalan Senthilkumar. Is there, sorry, one second. Yeah, is there any side effects occur if we consume such products?
So such products in the sense, in terms of commercial aspects, there are a lot of products with chemical preservatives or like other materials which are posing a serious harmful threat. But like speaking about our product, which is to be patented yet, has formulated only with the product or the naturally plant signaling molecules, and which is not posing any serious threat. Like products like or the signaling molecules, as I mentioned earlier, like melatonin or the GABA, has also been reported in animals as a neurotransmitter. And also, there are few patients who are prescribed by the doctors to supplement GABA or the melatonin or the spermidine or the putrescine antioxidant property, anti-signaling molecules in the diet of the humans. So, frankly speaking, use of our product like Biomax Plus will not cause any serious harmful effects in terms of human health hazards. So, these are all developed out of natural signaling molecules. So, these are are like eco-friendly in nature.
Okay. Before we end, let me check the participants in Zoom. Mr. Vasant, you would like to ask any question? If you are unable to speak out, you can post in chat box also. Okay, Mr. Siva, if you have further questions, you can post it or you can ask directly from the mic. Okay. I think nobody has further questions. So now we have come to end of question round. On behalf of agricultureinformation.com, we'd like to thank you, Mr. Shricharan, for the detailed presentation and answering all the questions in depth. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Thank you so much, ma'am.
You're most welcome. Thank you. Thank you everyone for joining the meeting.